Okay, so yeah, unexpectedly, I have to cut that other video. So the second video, we're going to talk about um, antibody production. We're going to talk a little bit more about how disease get transport. Now, the thing here, so this area talks a little bit about. It's kind of related to the pandemic situation. Um, Let me see. One of the things that is very common now, yeah, in terms of a pandemic, like if you study history of disease and study history of disease outbreak, I'm pretty sure a lot of you by now have, if you watch the news, have heard of something about the COVID-19, and they always talk about something about comparing it with the Spanish flu. The Spanish flu back in 1917. Now, the Spanish flu back in 1917 was actually, aside from the Black Death or the bubonic plague of Europe, that's probably sets the standard of what is a pandemic situation, a disease that spread around the world and caused a lot of death. Now, keep in mind, the Spanish flu was back in 1917. It was a very special circumstance back then. You're talking about World War One, when soldiers were fighting in trenches, and a lot of people coming from different places of the British Empire. You're talking about, yeah, back then we didn't even know what viruses are, and the most at best medication that we have was an aspirin. So in this case, we certainly don't know what virus is. We have no antibiotics, and there's no idea of how to isolate and protect from disease. So, as you can imagine, in situations like that, things can go bad really quickly. You're talking about a particular strain of the flu which severely attacks the young people, i.e., the soldiers who are in trenches. And next thing you know, the war is over, and all these soldiers have to go back to where they came from, and that's okay. So in this case, it transmit and bring the disease back to wherever country they came from. Yeah, it's gonna spread really quickly and go bad really quick. So if you think about that, the world of medicine is very different nowadays. We have antibiotics, we have antivirals. So in this case, uh, we're in a better place to deal with things. But in some ways, this COVID-19 is pretty deadly. If you think about, there's still a lot of people dying despite of modern medicine. So, anyways, coming back. So in this case, we since the Spanish flu, there has been a number of epidemic and pandemic, and has been going on pretty much. There's one every 10 to 20 years. The last major one before the H1N1 was back in the 60s or 70s, if I remember correctly. And so we have been actually, according to my professor when I was at McGill, we were long overdue for a pandemic. So the talk of disease spreading has been around for a long time, and that's one reason why nowadays you can get a flu shot for free. This program came up in the last 10, 15 years just because of that. So let's talk about disease passing species. So normally, a particular pathogen can only infect a certain type of host. Okay, certain host, a certain type of host. They're very species specific. Okay, so this here, polio, syphilis, and measles and gonorrhea. Interesting. Um, they're all pathogens that only affect human hosts, not so much in others. But when you're talking about disease that can go from other animals into humans, you call it zoonotic disease. Zoonotic disease. Thank you. if you think about the first part of the word zoo, that's usually a field associated with animals. And so in this case, you're talking about a disease that goes from animals to human. So here are some examples. Now, bubonic plague, I mentioned it already. Some influenza is from from birds. Now, you also have some influenza that are from pigs. So the swine flu from Mexico a few years ago, and rabies from dogs. So these are example of zoonotic disease. Now, usually when a disease go from animal to human, it's pretty bad. That's why those are the disease that we usually watch out for. Now, um, disease transmission. So, how are some disease being spread? So, for example, a couple of them. Direct contact. So that's physical association of bodily fluids. You have contamination. So usually you will be eating in the pathogen through your mouth. Maybe it's from food source or something like that. Could be airborne. So by sneezing and coughing. Now, this is the textbook definition. Now, I'm pretty sure nowadays you hear a lot of things about droplets and airborne versus droplets. Now, droplets and stuff like that, it's usually when you cough, you actually spray. Next time, if you need to cough or sneeze, go right in front of the window and do it. And you'll see that it gets pretty wet. Those are the droplets from your mouth. And a lot of times, the pathogen will be actually in the droplets itself. And then, of course, the surface, or if you unfortunately got sneezed on or coughed on, that's how you get it. But sometimes some pathogens will actually survive much longer in the air. So it's actually stay, some people saying, on the dust for a very long time. Those would be like true airborne type of disease. Now, vectors, you're talking about things that organisms transfer the pathogen without having symptoms themselves. So the rats for the bubonic plague, that's an example of that, because actually the bubonic plague is, it's, if I remember correctly, a bacteria that infect the flea. I think it's a bacteria or parasite that infects the flea that is on the rats themselves. So here are some diagrams of other types of zoonotic disease. So that's just a little blurb here, talk about disease transmission. Now, antibodies development. Now, this here is very similar to what we talked about in our previous lesson. So let me see. So this diagram, I put it on our board. This is actually very much, if you think about not cross contamination, talk about disease transmission. This is kind of the practical aspect of what you need to do, and that's what I had to do when I first started taking microbiology. They trained us on it, so you're being mindful and not do those things yourself. No, the handout. Where's the handout? Right here. So this here, go back to a little review. So in SL material, so this is what you learned about the process of antibody production. So pathogen got engulfed by phagocytes, display or present the antigen of the pathogen on the surface, activates the complementary T cell. T cell stimulates the B cell. B cell makes clone of itself through mitosis, either plasma cell or memory cell. Okay, so that's what you learned in SL. Now we're going to learn a little bit more detail. What actually happens? Now, as I said in previous videos, this material itself is very much second and third year university material, at least in Canada. So in this case, there's actually much deeper than that, but this is the version that IB chose to teach in high school. Okay, so it's still pretty deep, but it is, keep in mind, a simplified-ish version. So here, non-specific immune cells called phagocytes. Phagocytes, you're engulfing pathogens, present the antigen fragments to specific helper T cells. So this is similar to what we talked about previously. I'm trying to find a diagram of that. So this part is not presented in this diagram. It's not shown in this diagram. But here it talks about clonal selection, which I kind of talked about in the other video. So when activated helper T cell, it releases cytokines. Now, for those of you who watched the video that I suggest, the coronavirus playlist, there's one video that was presented mainly to talk about the pathogenesis, basically the disease development. And the guy in that video talked a lot about cytokines or interleukins and stuff like that. So cytokines is a type of chemical signals. There are many of them. So when we say cytokines, that's a broad general term. Okay. So the helper T cells will release cytokines which signal specific B lymphocytes. Okay. So the specific B lymphocytes may copy on themselves. That's clonal selection. Now this diagram here, it's not the best. Okay, this is diagram again is not the best. I will go back to what I shared with you on the handout, the Wikipedia page diagram. This one is much better and much more clear. Okay, so in this case, the specific B lymphocytes may copy up themselves. So here, basically within this step, so you have B cells that are made in the blood, in the bone marrow. Some of them are eliminated because they are specific to own self antigen. So the MHC MHC bodies, that's what they are specific to. So they have to be eliminated. So they're not going to be attacking your own body. So now you have these B lymphocytes. Now, imagine you have helper T cells, and only this particular kind that is specific to the antigen, the foreign antigen. This will also be activated by helper T cell. Hence, this becomes clonal selection. Creates clones of itself. Okay, creates clone of itself. Some will make memory B cells. Some will make plasma cells. Okay. Yeah. So here you have 3 different kinds of B cells. Only this one is specific to the foreign antigen. So this one get activated. Yeah, this one get so the helper T cell will release cytokine to signal this particular B lymphocytes to make more of itself. So as you can imagine, if your if your helper T cell is compromised because of HIV, then this pathway of making antibody will be limited or also compromised too. Okay. Pause right here. Do you have any questions? Okay. It is a concept that we talked about a number of lessons. However, it is, as I said, second, third year university level stuff. So I'm making sure that you are repeat a number of times and say it a number of times. Okay. Now, because each pathogen has several antibody cell or B cell can activate it, this is called polyclonal response. So it is a good thing to have a polyclonal response. Also means if you think about it, that's the reason why when you're sick, your first few days, you're really tired out simply because your body is using a lot of energy to do this. Okay. Now, antibody, this here, the um, the mnemonic panic. Okay, so what do antibodies do? Now, in SL, you learn that it just binds to the pathogen. It either neutralizes it on its own or it's become tagged for pathogen to be to eliminate it. Now, you're in HL, so in this case, you have to learn a little bit more. So first of all, so some toxins are become insoluble or precipitate out, so will no longer remain in the bloodstream. It will just fall out and then get eliminated. So actination. So in this case, some of the pathogens will be clumped together. So A can be easy removal, and B, it might make it harder for those pathogens to invade the cell. Okay. Neutralization, antibody attachment stop the toxin from entering and affecting cells. So if it is a toxin, now toxin is actually just a chemical that is created or produced by the pathogen itself. So the pathogen might be dead. Let's just say for some reason it's dead, but you might still have toxin in there. So those toxin, your antibody can attach to it so that it can either stop it from going inside the cell or affecting it. Okay. Inflammation, so trigger inflammation response, complement activation. So sometimes this is a part where sometimes the antibody will eliminate the pathogen itself. Basically, it punch holes. Okay, complement protein perforates membrane. There's some proteins called complement proteins. It'll gather together to where the antibody is and where it tags the pathogen, and it will punch holes into it. So here is some diagrams explaining it. Okay. You can read that. Any questions? You just kind of have to know this part and memorize it. Okay. Like Tracy, for example. Now, in terms of in terms of antibody and immunity. So the reason why you have immunity in the first place is because you previously have exposure to a certain type of pathogen. Okay, so the first time you ever meet that pathogen or this particular antigen, you call that the primary immune response. So right now, most of us have not encountered or been exposed to COVID. So if we get COVID or exposed to COVID or get COVID, we will be experiencing a primary immune response. Now, those people who got COVID and thankfully recovered, those people in their system will have memory cells now. They have memory cells now. Again, I kind of talked about this last lesson. It takes about 2 weeks for the primary response to kick in. B cells, so you have memory cells. Now, if those people who recovered from COVID encounters COVID virus, and presuming that it has not mutated, again, because of the duvet? Yeah. Okay, hold on for a second. Bless you. Yeah, I'm back. Where am I? Okay, so those who recovered will have memory cells in them. Now, the memory cells, one of the big difference between memory cells and plasma cell is this: plasma cell usually die off in a few days. Memory cells stay in the body for a long time. So when the second immune response happens when the people who so if those people recovered from COVID-19 encountered the COVID-19 virus again, assuming that has not been mutated because it can, so it's exposed to the same antigen. Okay, so in this case, the memory has been established. Will make the antibodies faster. So this response is much faster and the antibody, and the response will be much larger. So if you look at the antibody concentration in the system, it peaks a lot faster and actually produce a lot more antibodies. And actually, the antibodies will be attaching to those antigens a lot faster. So the notes here says usually kill within minutes. So this person's said to be immune. So active immunity is when you act the immunity because you produce the antigen by the organism itself. It is naturally acquired or artificially acquired. So naturally acquired means you got the pathogen in your body and you survived, or you taken a vaccine that's artificially acquired. Now, passive immunity. This is a part about you, the immunity from getting the antibody from a different organism. So let's just say this could be naturally acquired by breast milk or placenta. So the baby in the womb, it was protected by the mother's antibody. And also when the newborn baby, when they're first born, when they're first born and they started breastfeeding, they also have the antibody from through the breast milk. Now that's only good for about 2-3 months or so. Later on, they will need to be vaccinated and whatnot. So, or artificially acquired. So you can have injection of antibodies. And last week, this is again interesting because we're living in this COVID-19 situation. This is actually interesting because last week, FDA, okay, let's see, FDA, I still have that here. So FDA, what they have, when is this? Yeah, March 24th. So that was last week. What they're saying is that they're going to be trying out COVID investigation of COVID-19 convalescent plasma. So essentially, what they do is they're going to get basically blood from people who recovered from COVID-19. They're going to take that blood, spin it down, take the plasma, which contains the antibody, and then inject it into people who are in serious conditions. So as you can see here, for use in patients with serious or immediately life-threatening infections. So in this case, that's what they're doing, or that's what they're experimenting right now. Traditionally, this works. Okay, in fact, during the Spanish flu, when people have no idea what a lot of these things are, that's what they were trying to use. They were using plasma to try to put it into from people who recovered, put it into the people who are sick. The downside of it is that these antibodies don't last long in the system, so you have to constantly replenish the plasma. Okay. So here's a list in terms of what the different kinds of immunity and what they're called. So I was trying to talk about antibody production. So antibody production and vaccine. So that's where I am right now. I'm going to stop because this has been also a pretty long video. If you have any question, please make sure you write that down. I'm going to make the third and last video about vaccine, okay?